Thank you. Uh, Thank you for the kind invitation for this session. And I think this is a very good idea and a very good tradition that has been set. And I thank Dr. Ganguly and the organizing team for inviting me as chairperson for this very important session. And I would like now to invite uh, the first speaker, who is extremely eminent person, Dr. Firdausi Qadri, who's a senior scientist in the infectious disease division and also the head of the mucosal immunology and vaccinology unit at the famous ICDDR in Bangladesh. She's extremely well published, and obviously, these publications have had a huge impact because she is extensively honored with some of the most prestigious awards that are there for the taking, including the CNR Rao Prize, the Raman Magsaysay Award, which is the Asian equivalent of the Nobel Prize, and also very recently the Swadhinta Award, which is a very important award from Bangladesh. Thank you. So, Dr. Firdausi, may I request you to begin your talk?
Thank you very much. Good morning, everyone. It's a pleasure for me to be in this meeting, though virtually. I'm sorry I did not get my visa and I could not come. I was looking forward to coming to Chandigarh and meeting everyone face to face, and Professor Ganguly, Neelam, and of course, Professor Talwar, and everyone else. But I'm going to try to give my message on the importance of probiotic, not so much of probiotic, but the gut microbiome. As we are aware, there are many microbiomes in the whole body and trillions of bacteria, more than the human cell. And our emphasis has always been more on gut because of the interest in enteric infection. So in my scope, I will try to cover areas that we have been involved in and which are very important for moving forward for effective vaccines and effective treatment. So because I've been working on cholera for 30 years and more, I will try to relate this to cholera, my talk to cholera, but also to other infectious disease. Since the audience over here will not know what we cholera people know, so I'm just going to go back into a little bit of the historical events. And so we are in the, it's an ancient disease, cholera. And in our Indian subcontinent, we do not want to talk about it because it's a disease of poverty. And there have been many epidemics and pandemics, and 6 of the 7 pandemics in the last 200 years originated in South Asia. And so we have this term Asiatic cholera, which is sort of a derogatory term for us. During the first pandemic, first cholera case was actually reported from Jessore on the border of India in August 1817. And since then, cholera has claimed many lives. In our country, we have many diarrheal cases every year, and 66 million people are at risk at cholera. This is our estimation. And at least 5,000 die yearly at risk. And I'll come to the next slide to talk about what I want to say. So we are living in an era of extreme natural and man-made turbulence, climate change, pandemics, floods, tornadoes, earthquakes, tsunamis, and humanitarian conflicts. So the problems we are facing now is rising sea level, which is affecting agriculture, salinity, and many other things. We are also having lots of cyclones, floods, and urbanization. In the last 2-3 years, we have faced extreme floods and also cyclones, but more floods and also droughts in certain areas. And droughts and rising sea levels have led to a lot of salinity in our areas. And then you see more and more cholera patients every year. And I come from the ICDDR hospital, which is actually known as the cholera hospital in Dhaka. And there only we see 20,000 cases at least every year, and more during other times. And In order to prevent diseases, there are many strategies, many pillars in the global roadmap to decrease cholera, and there are many pillars. And it's not only vaccines, but it is also WASH and also surveillance. Very good surveillance that will help us move forward. But those are not happening so quickly. So I'll just give you a little bit of an example what we've been trying to do. We have tried to use the cholera vaccine, the oral cholera vaccine, which was produced in India but has stopped production now, Shancol. And we have been able to show that if you can use the cholera vaccine, if you can use the cholera vaccine under EPI conditions, like just dole it out and give it to people using the mechanism of the EPI, the National Immunization System, you can do a good job. And so we have the red areas over here in the map of Dhaka. We have vaccinated people in 6 areas, and we've also given WASH to the people, chlorine pumps and water areas where you have water and bottles of soapy water. And then we've shown that you can prevent cholera to a large scale, but we know that we have a big population, and according to the latest estimates, we have about 172 or 73 million doses of vaccine required, and the global shortage of vaccines is acute. And so we are trying many ways of trying to fill the shortage, especially when Sanofi has stopped making vaccine in India. We have other companies coming up, and so we are looking towards oral cholera vaccines made in Bangladesh and also in Korea, which are coming about to meet the shortage in the Gavi and in the cholera stockpile. But when we talk about all these things, we must remember that there are other things happening. So we have done studies to look at oral cholera vaccine protective efficacy. And whenever we look at different age groups, we find that, the vaccine in younger children doesn't do so well. Like it's 16% effectiveness. Whereas if you look at children above 5 years, it's 63%. If you look at children over 15 years, people over 15 years, it's 56%. And it prevents against 63% against severely dehydrating cholera officers. I'm just talking about the oral cholera vaccine. So then the sum total of this is that oral cholera vaccine is efficacious in older children, and it does, but not in younger children. This is not the story of oral cholera vaccine, but is the story of all vaccines that are oral. Oral cholera vaccine, oral polio vaccine, oral rotavirus vaccine, and so on. And even the oral typhoid vaccine. So, I mean, there is a big problem, and why are these problems there? And so then we come to the subject of this conference, which is the microbiome. And I will talk about microbiome mostly on enteric infections. And so Vibrio cholerae has to be ingested in very large amounts to cause disease. And when Vibrio cholerae survives the acidic gastric environment, it enters into the small intestine, and into the small intestine does not enter. It tries to enter the gut lumen, and it interacts with bile and also with mucus. And these are signals to Vibrio cholerae to express virulence factors that cause symptomatic disease. Mucus coats the villi and acts as a diffusion barrier, and Vibrio cholerae uses flagellar motion to traverse the mucus and get into the closer to the gut. During this journey, Vibrio cholerae encounters resident other gut microbes, the microbiome, and their metabolites. And after reaching these are the pictures. After reaching the intestinal epithelial crypts, the Vibrio cholerae forms biofilms. Look at the biofilms over here to adhere to the epithelial surface. And when encountering the mucus layer, the type 4 secretion system is activated. This system is very important and to make Vibrio cholerae in contact with other neighboring organisms in the gut. The type 2 system is also very important it is suppressed by metabolites of cholic acid, but autoinducers are produced by some commensal gut microbes and can induce quorum sensing. The presence of autoinducers then goes on to the valence properties, and then the bacteria can grow, and also, but there are also bacteriophages that can kill the bacteria over there. And so the gut microbiota is a factor influencing susceptibility to cholera. So there are many experiments and data that show that Vibrio cholerae is able to infect animals more easily after antibiotic administration, which means after the gut microbiota has been cleaned. Increasing recognition of host microbes' role in human health, as Professor Talwar was saying, not only in the gut, but also in the vagina and also in the respiratory microbiome, all over the body, all the microbiomes which make over 39 trillion microbial cells. So the preliminary, I'm going to talk a lot about data on cholera cohorts that we have been following, and the differences in the gut microbiome of cholera patients, which we call the index patients, and the household context, who become infected or who don't become infected. The fact is there is one person coming with full-blown cholera to our hospitals, there will be at least 3 other people infected, one maybe full disease, maybe just infection, mild diarrhea, or also maybe increases in immune responses, which we can measure by vibrocidal antibodies. So they become infected. So we have identified bacteria associated with protection or susceptibility to Vibrio cholerae in a household contact sample. I will talk about these. And are these bacteria present at the site of infection during control? We don't know. What are they doing? We also don't know. So this is a design that I will talk about. We have had about 4 or 5 cohorts that we've been following. Patients who come to the ICDDR hospital, which is at the very beginning. We call that day 1, the presentation of index cases. And then we follow them up with, we go to their homes at day 2, and then we follow them up for 10 days, taking stool or rectal swab from them. And then again, we go to their homes at day 30. We take rectal swabs or stool, but we also take blood from them at 3 time points. By that, we look at the vibrocidal antibodies, and more recently into more effective studies like parameters such as the memory B cell responses. So contacts with positive rectal swab specimens developing watery diarrhea are categorized as symptomatic, and those without diarrhea are considered as asymptomatic. So while doing the studies, we obtain a lot of mucosal samples, not only the stool, which is our very important specimen for mucosal responses, but we also look at the duodenal microbiome. And we have permission for studies to take the duodenal biopsies, and from that we can also follow up. So we have, in one of our studies, we have taken biopsies from patients when they first come to the hospital and have been rehydrated, and that is at day 2. And then we follow them up with at 1 month, 3 months, 6 months, and a year. And we look at compare the microbiome and also other immune responses at the day 2, and then follow up with the biopsy results by phylogenetic relationship and look at the microbiome. So a predictive gut microbiota of roughly 100 bacterial taxa discriminated between contacts who developed infection and those who did not. So that is very important. The susceptibility to cholera was associated with depleted levels of microbes, because the Bacteroides, which is one of the 3 important phyla in the microbiome of the gut. By contrast, a microbiome associated with cholera by our modeling framework showed that Paracoccus aminovorans promoted the in vitro growth of Vibrio cholerae in vitro studies. Gut microbiota structure, clinical outcome, and age were also linked. So this is a graph to show how the Paracoccus aminovorans spent supernatant were compared with Vibrio aminovorans, which is non-pathogenic, and we looked at the spent culture filtrate of both. And we found that yes, this the Paracoccus aminovirus does have a contribution to the CFUs, increase of CFUs, or the optical density, which can be translated to CFUs per milliliter in the gut. But also we've looked at changes in gut microbial succession in the weeks following Vibrio cholerae infection. And we've looked at two cohorts, cohort 1 and cohort 2, and looked at the percentage of cholera in the two cohorts. So the bacterial OTUs were collapsed at the genus level. So we saw highly conserved genera and further grouped them according to the dynamics. So you can look over here at infection stage, at which we can expect it's only Vibrios. At early stage, it is Enterococcus, E. coli, Streptococcus. At mid stage, it is Bacteroides. And then at late stage, you're coming back to Prevotella and Roseburia. And then if we mix up all the swabs and took it together, then we get Anaerococcus, Corynebacteria, and all these other important bacteria in the specimen. So it does show that the gut becomes depleted of other bacteria except Vibrios at the initial stage. And so looking at the duodenal microbiome of patients, we have also looked at, and we've shown that in the very first phase, it is Vis-Stivna Valley, and then at Firmicutes number 2, then it becomes Firmicutes again, but a different species, and then it becomes Proteobacteria. So there are more aerobes and less anaerobes during acute phase over here. We've also looking at Vibrio cholerae infection and seeing that does it help us, does the metagenomic features help us better than clinical and demographic features? So we did random forest prediction and over here and looked at the status applied to 7 clinical and demographic features and compared with all species and all gene families. And we found that the gene families clearly support superior predictions, and adding clinical data did not improve the predictions based on microbiome features alone. And these results also demonstrated that gene families present in the gut microbiome at the time of exposure contain more information about Vibrio cholerae susceptibility than species level of clinical and demographic data. That itself is very, very tantalizing, because we depend so much on the demographic and clinical data that when we look at patients. And we've also looked at as I said, we have household contacts who are protected, who are asymptomatic, uninfected, and infected. And we saw that Prevotella species were a large contributor to pathways more prominent in uninfected controls, long-chain fatty acid metabolism, gondoid and vaccinetic acid were prominent pathways among uninfected controls. So those are very important data that can make us find solutions. So at the time of oral cholera vaccination, at the baseline, the microbiota of study participants predominantly, this is about vaccination, composed of phyla, Acetobacteria, Bacteroides, Firmicutes, and Proteobacter. Then we looked at further analysis of beta diversity using principal component analysis, and to see if there was any overlap from the non-responders and responder bacterial communities. But related to the non-responder and responder participant microbiotas at the time of vaccination. But I think we were not able to find anything like that. But so this is on cholera. But just before I move away from cholera, I would like to tell you that we did a study to look at oral cholera vaccine Ducorol and the effect of a probiotic that's actually made by Yakult. I saw in the morning session that there are people from Yakult, and I remember Professor Nishibuchi from Japan, with whom I worked to look at with my group to look at it was Bifidobacterium brevi, and if it had any improvement in the response of young children to the oral cholera vaccine, but we are sad that we did not find anything remarkable. So the role of, I will move on to other things that are being happening around me in ICDDRB, looked at the role of gut microbiota and disease severity and treatment outcome in Guillain-Barré syndrome. We know that Guillain-Barré syndrome in our setting or many other settings is 50% due to Campylobacter jejuni. And we found that gut microbiota was compared between GBS at baseline and 6 months after entry into the study. And we looked at the GBS at 6 months. So this was the gut microbiota dysbiosis is present in GBS patients compared to healthy controls. These are healthy controls, and this is the GBS patients at entry and GBS patients at 6 months. So significant alterations in predominant gut microbiota were observed between acute GBS and immunoglobulin therapy treated GBS. So that is something important that we must understand. And we are actually doing a lot of work on GBS, and we see the alteration of gut microbiota observed with acute GBSE, and that after 6 months of follow-up, it does go up, the gut microbiota. And then also there have been other studies work on role of gut microbiome in Guillain-Barré syndrome. And at the acute stage, Streptococcus was overrepresented. Over here, and you can see there was more of Streptococcus. And this is a list of gut microbiota identified as potential biomarkers of GBS induction. So these are food for thought for how we move about with the disease, which actually is very difficult to control in settings like ours or any other setting in the world. And then there's more work on dysbiosis of microbiome due to Helicobacter pylori. We are all infected with Helicobacter pylori most of us, I am quite often, by Helicobacter pylori. And the principal coordinate analysis showed that Helicobacter pylori infected samples creates a distinct cluster than the control samples, indicating significant changes. Also, H. pylori infection reduces diversity and robustness in the microbiota. And the bar plot shows that there's difference between controls and between H. pylori infected people. And the healthy tissues show diversity and richness of other microbes, as a major proportions are E. coli, Plasmodium, and Streptococcus pneumoniae. But in the most abundant species in GBS is not these other bacteria. So the dysbiosis of microbiome due to Helicobacter pylori, more work that has shown shows a mutualistic interaction with other microbiomes, positively correlated with Helicobacter hillmani, Acinetobacter baumannii, one of the major phylums, and in the microbiome, and the Phylococcus hirokoshi, and also inversely correlated with Bacillus thuringiensis. And then E. coli, Bacillus, and Chronobacter are showing positive correlations and with Bacillus thuringiensis, but not a negative correlation with Helicobacter pylori. But also, we cannot leave typhoid bacteria away from here, showing that although it causes both the mucosal, but mainly a systemic infection, in Salmonella typhi chill models carried out not in Bangladesh, but in Oxford, it has been shown that there are two transcriptomes, that in infected and non-infected samples did not show any significant differences. However, in transcriptomic analysis, two distinct communities were found in the transcriptomics community. One of them was enriched with methanogen microbes, and the other evenly dispersed. The highly reduced environment of gut methanogens may mitigate the effect that is produced in GBS, in Salmonella infection, that alters the signals by triggering a cascade of virulent factors for host invasion. And we can see that there is the adaptive immunity that is under play, and cell surface receptors such as CD86 and CD197, and monocyte-derived cells. So what about nutrition? We are in a country where we see a lot of stunted and undernourished children, weight for age, difference stunted, weight for length for age, but also shows high weight for AIDS Z-squares, but there are studies that being carried out in the community. In Professor Tahmid's and his team, Mahfouz, they're working on this and working with the University of Washington, and they've shown higher weight for AIDS Z-scores when two complementary foods were used. The supplementary foods were used, the microbiota direct complementary food or the ready-to-use supplementary food, RUSF. And you can see over here that the complemented, the microbiota directed complementary food, MDCF, had a better effect than the commercial RUSF. And then the other data that came out was that 21 microbes had positive correlation and 2 microbes indicated negative correlation with weight for length Z-scores. The amplicon sequence variants over here showed that in the normal gut microbial community in Bangladesh children, and the amplicon sequence variants show associations with weight gain in gnotobiotic pig. And so these are gut microbial samples that have been tested in gnotobiotic pig. Then the abundance of weight for length Z-square associated taxa were significantly greater in the microbiota of children whose diets were supplemented with the MDCF2, but not those who were supplemented with the commercial RUCF. So what else was found was that Prevotella and Faecalibacterium showed highest increases, while Bifidobacterium species exhibited the greatest decrease, and microbes show positive correlation with 70 proteins due to MDCF2 complement. The MDCF2 produced a significant enhancement in the rate of change in abundance compared to unsupplemented children, but RUCF did not exhibit such significant difference. And after 1 month of cessation of the supplements, the weight for length Z-square associated amplicon sequence variants began to fall. At the same time, the weight for length Z-scores started diminishing. So those data can tell us which proteins, which bacteria that need to be added, isn't it? So what has come out from this is that if you look at the probiotics, which is the talk today with the conference that is mostly centered on, and I was looking at the number of probiotic products. I Googled and found there are 4 billion products available, but we know that all of them don't work. But in our natural diet, we have yogurt, which has billions of bacteria that can support us in our fight against many of the diseases in our countries. And so there are 2 bacteria, Butyvibrio and Megasphaera and others, that have been studied in the mouse model of enterocolitis. This mitigates weight loss, mucosal damage, and diarrhea. And the supernatant from Butyvibrio fibrosolvens culture inhibited growth of Campylobacter species in vitro, and also given to mice prior to and during Campylobacter infection, and decreased diarrhea and mucosal damage. So that's very encouraging. And Megasphaera induced recovery of mucosal atrophy in weaning piglets. And then I've looked at the literature that's available. Toxin binding, GM1 expressing recombinant probiotics may have potential of binding cholera toxins and other related enterotoxins. I think maybe the LT toxin of enterotoxigenic E. coli. Also, nutrobiotics may have a role to play in improve nutritional status. So in conclusion, I would say that the gut microbial taxa influences the response and take rate of vaccines, because we don't see vaccines working well in our countries, and also natural infection. Even in natural infection, the response is poor. And we have data to show the antibody magnitude of antibody responses, the memory B cell responses, the mucosal responses are all stunted in children and in our settings, and the consequences of diseases. Specific microorganisms and gene families such as iron processing and metabolism are more prominent in the organism associated with the microbiome of uninfected persons. In my feeling is that these studies demonstrate that manipulating the gut microbiota to alter the course of cholera, just a case, but also other diseases, and malnutrition may be possible. In our countries where we will not be able to have wash and general nutrition levels increase quickly. Advancements in understanding immunity and susceptibility to Vibrio cholerae increases global commitment to disease prevention. And I also feel that I'm a vaccinologist, and I feel that better nutrition status of young children may be achieved by interventions to improve disease prevention and vaccines. And vaccines may not be the only preventive method. I've been working in children and with all these oral vaccines, and I feel that maybe we have to look for the probiotics and also other, in addition to vaccines. I wouldn't say one or none, but a combination of all. And with that, I finish my talk. And I again thank Neela for inviting me and Professor Ganguly. And I'm sorry I cannot be here. I would have loved to be in Chandigarh. And I thank my colleagues working in Bangladesh and everywhere else. Anavil, who was a fellow in our lab and gone back to Washington University. And then we have from ICDDR and also from the Harvard Medical School, where together we are looking at the household contact, cholera patients, salmonella patients, and vaccines, and for all pathogens that we can think of. And there is so much more to do. Thank you again.
Working on? Yeah, now it's working. So, thank you very much, Dr. Firdausi Qadri, for a very, very comprehensive and elegant keynote address. We definitely miss your physical presence here. That would have given a chance to many participants here and experts to interact with you in person. What I really appreciate is the public health perspective and its connect with the detailing of gut microbiome in those infections which are very, very common in developing countries. You spoke about cholera and GBS, H. pylori, Salmonella typhi. I think these are the things which we see very commonly in India as well. And your excellent work in terms of detailing the gut microbiome in these conditions, and your words about that the complementary microbiota directed complementary feeding definitely has a beneficial effect on the gut microbiota, and there's huge scope for nutrition intervention to come in the forefront. And being a nutritionist, I think I'm very heartened by these concluding remarks of yours. And we always vouch for preventing rather than curing the disease. And these are all the conditions where I think in developing country, we can do a lot by preventing some of these very, very common enteric infections. So thank you very much for that excellent keynote address. I think all of us have really benefited. I just wanted to ask you, Dr. Qadri, would you be staying on for the next two lectures and then take the questions, or would you like the questions to be asked now?
I would prefer the questions to be asked now because I don't know about my internet.
Okay, fine.
Yeah.
So maybe we could have one or two questions. Yeah, please give the mic. Please give the mic to in the audience. वहाँ नीचे सर को दीजिए प्लीज. Yeah. Dr. Qadri, do tell us if you can hear the person who's asking.
I can. I haven't heard, I cannot hear any question, but I can hear you perfectly.
And then ask.
So myself, Javed Agrewala from IIT Roper, Punjab. My question is do you think then the efficacy of vaccine is modulated by the gut, and that supplementing that particular gut microbiota is ultimately going to increase the efficacy of vaccine?
I certainly think so. I mean, first of all, we've been working on oral cholera vaccines for a long time, and it has been, we've concentrated on oral cholera vaccines because that's gives us 60% protective efficacy in us and effectiveness in our settings. But when we talk about young children, we always fail. I mean, it's not that it's not something that has not been noticed earlier, but the oral cholera vaccines or oral rotavirus vaccine or oral polio vaccine, there is a deficit. But for oral cholera vaccines, even a study that we just carried out and we've submitted, that's going to be published, we've shown that in a large field trial in Bangladesh, when we were trying to prevent, in a preventive vaccine trial, and we looked at the effectiveness of the vaccine, in the under fives, it was only 16%, whereas overall it was like 70%. So, I mean, this is something that we need to think about, improving vaccines, maybe to some extent going back to parenteral vaccines. Also, those things are also in the way, on the process. But for younger children, we have to think about something else. And
Dr. Qadri, we are really missing you over here. There is one more introduction to Dr. Qadri. She is popularly known as APA. That means body band. So, because he has this kind of a personality, gives umbrella to large number of young investigators. My question was this, that your data in GBS is very, very interesting. We have now infection going on in Mumbai area,
Yes.
Pune area, where children are dying with the GBS kind of symptoms, who are infected by an organism. So during the COVID infections, there were some GBS-like things which came up from the neurological. Your work shows that this could be used to ameliorate the symptoms of inflammation in GBS effectively. This data is very, very powerful data which you have shown.
Professor Ganguly, I always expect you to ask the hardest questions, the most tough ones. And I'm missing my presence over here because I won't be able to talk to you and the others and learn a lot from you all. And so actually GBS work, Dr. Zohir from ICDDR has been working on using immunoglobulin, plasmapheresis, and all these things. And this is part of his work. And I think even here, there is a role for the gut microbiome to play a role, definitely.
Can I ask a question, Dr. Qadri? I'm
Yes,
Dr.
sir.
Sadhana. Yes, I'm
Yes.
very intrigued, and obviously we are all into the era of gut dysbiosis being at the back, probably at the center of all many of the autoimmune disorders. So while you were talking, it just crossed my mind that since you are working on nutrition as well as the gut microbiota manipulation, are there any studies or is there any evidence that particular kinds of foods can replace the bad bacteria by the good bacteria, and would those diets be in the pipeline? For example, we deal with a lot of inflammatory bowel disease, and fecal microbiota transplantation is one of the modalities to deal with it. But in addition to the good bacteria being used to manipulate the dysbiosis, symbiotic approaches, that means the use of particular diets to more effectively counter the dysbiosis. Is there any evidence in literature or in your personal experience as to that?
From my experience at ICDDR, I would say that the first thing that I want to tell Professor Ganguly, that Dr. Zohir at our GBS works on GBS. So it's not exactly my work, but our work at ICDDR. What you asked, Professor Sadhana, is very important. Dr. Tahmid and his group, they are basically very important nutritionists in the country and globally. They have been working on microbiota dietary complementary food, and it's made of chickpeas, or made of chickpeas or lentil, and they've actually patented products, and they have shown that they've just finished a trial in the Rohingya population, and have shown that the nutritional status of children, stunted children, malnourished children, improves when they're given these microbiota directed food. And they also are coming up with lots of other microbiota data. And so we do have solution for the severely malnourished children, the SAM children, severely acute malnourished children, who we see at the ICDDR hospital. When they come, we put them in a special ward, and when they're given this, or you can just put lentil, rice, and some other vegetable together, you will get, we will be able to improve the diet in a month, or now that we have a product, actually. It's not any other RUSF supplementary food, but a local, acceptable RUSF, MDCF that is actually working. And so we are very looking forward to this for our severely acute malnourished children.
If I can just add to what you said, Dr. Qadri, this complementary microbiota directed complementary feeding, microbiota directed complementary feeding, and the composition, what you said. Essentially, if you look at the ingredients, it is giving all the bioactives, whether it's soluble fiber or it's protein or resistant starch. So, if you go deeper into the combination which has been picked up, it is providing all the food which is going to favor the growth of the desirable microflora.
Yeah,
so
exactly.
and plus it's going to favor the growth. So I think it's a 1+1 kind of a synergy you get.
Yeah, also you're right, because this is now working. I mean, it has worked in our hospital, but now it's a patented product, because in a malnourished and in a poverty-stricken, wash-depleted setting, you cannot ask people to make these things all the time. But you have a small sachet that you can use to feed children, that would be very good. So we did have a very big dissemination on Thursday, and everybody was very excited. And this whole team, Dr. Mahfouz, Dr. Tahmid, and working with Washington University, they are doing this work and really very remarkable. That is going to be applicable for us.
Any more questions? Yes, okay. Dr. Kea, please give the mic to Dr. Kea.
Hello, it's nice to listen to your talk. I'm Dr. Lahiri from India. I'm a pediatrician. And as you rightly said, we had heard of Dr. Tahmid's lecture also, the previous 2 turns. And it is important that unless we control MAM, because it can lead to SAM, that is moderate to severe. And the point is that the lentils, the fish, the rice, I think that diet which has been proposed, in India, of course, we are having the midday meal program with khichdi and an egg. And that itself is causing a lot of plus points. And I think we have to severely think of nutrition in not only the children, the mothers. Of course, the father's income of the mother is important because she has grown up malnourished. And there are decades of malnutrition which you can't control. Just feeding the mother last few months of a pregnancy is not going to produce a healthy child. And hence we are having lectures on that. I don't want to dilate on that. But that is one comment I would like to have some answers from you if you can.
I think, of course, in the work that Dr. Tahmid is carrying out with his colleagues, Dr. Mahfouz and others, they're looking at SAM, because that is affecting children and leading to death. But I'm sure that there will be data for MAM children also. And it is very crucial that we find other ways. So this is the Shonali 1, Shonali 2 that they're working on will be less expensive because of the ingredients, but they will be able to activate the gut microbiota, the right gut microbiota. And so there are many other additional studies being carried out to look at the gene families, the different microbiota that is increasing, and also, as I said, the autobiotic piglet also. And so I think those are important, but I agree that the women have to be treated from not only at the last trimester, but throughout, because we we know that in the uterine, the changes that take place can be irreversible. So I think this is a country that our countries have to, this is a
The quick comment
fact.
is that having cholera is, as you said, it's not good news for any country. And we have considerably reduced it. There were pockets in Mumbai and India wherein cholera was seen. But I think the wash technique and other things are so very important, and we have to look into that matter. Thank you.
Thank you so much. But in Bangladesh, we still have a lot of cholera. In places of, especially in high density urban subpopulations.
Yeah. So thank you for that question. I think Dr. Ishi, yeah.
Yeah,
Please go ahead.
Ishi Khosla from New Delhi. I'm a nutritionist. Just a question on vitamin D. Has there been any kind of a intervention or investigation on vitamin D status and supplementation for mucosal integrity?
Not in terms of not in my knowledge in terms of enteric infections, but in terms of osteoporosis and things like that. And I would think that vitamin D is very important, not only for osteoporosis, first of all, in modulating the adaptive and the innate immune responses. So that is very important. But I think we did a study with vitamin A and zinc and oral cholera vaccine. And we did find a small effect, but not vitamin D. But it was zinc has a very profound effect in promoting the immune responses and increasing the immune responses in young children. We have looked at it not in cholera, but in enterotoxigenic E. coli, which is just a very big cause of acute watery diarrhea and epidemics in our country.
Mm-hmm.
Thank you. Thank you, Dr. Qadri, for an excellent interaction with our participants. And it's the life cycle approach, which we are all emphasizing. I'm very heartened to say that Government of India is going very aggressive on this life cycle approach. And, the latest announcement by the Prime Minister on Suposhit Gram Panchayat is a step towards that, where we are wanting to reach out to Anganwadis to cover the young children, pregnant women. And also there is an endeavor to reach out to adolescent girls, which is a preconception phase before pregnancy. So I think a lot is going on in our country as well, but it's very heartening to see the good work coming from Bangladesh, and globally we are talking the same language. So thank you once again for your time and your presence here.
Thank you again. Thanks a lot.